Hello and welcome to my YouTube channel. Hope you all will be fine. In today's video, I am going to discuss about the enzymes. Important inorganic cofactor along with their respective enzyme include ferric chrome oxidase and zinc carbonic anhydrase and magnesium glucose 6 phosphatase. Many enzymes contain vitamin as their cofactor. For example, nicotinamide adenine dinucleotide contain nicotine amide vitamin and thiamine pyrophosphatase contain vitamin B1. While naming the coenzyme, suffix ase is added to the name of the substrate on which the enzyme acts. For example, urease, sucrase, cellulose are the enzymes which act upon the substrate urea, sucrose, and cellulose respectively. Classification of enzyme. The Commission on Enzyme appointed by the International Union of Biochemistry IUB classified enzymes into 6 main types. So let's discuss about the classification of enzyme according to the International Union of Biochemistry. Number 1, oxidoreductase. Number 2, transferase. Number 3 is hydrolase. Number 3 is hydrolase. Number 4, lyase. Number 5, isomerase. On number 6, it's ligase. So first of all, I am going to discuss about oxidoreductase. These enzymes catalyze oxidation reduction reaction. Common examples are oxidase, dehydrogenase, and peroxidase. On number 2, we have transferases. These enzymes bring about an exchange of functional groups such as phosphate or acyl between two compounds, for example phosphotransferase, etc. On number 3, we have hydrolases. These enzymes catalyze hydrolysis. They include proteases called proteolytic enzyme. After that, we have lyases. These enzymes catalyze the addition of ammonia, water, or carbon dioxide to double bond or removal of these to form double bonds, for example phosphoglycerimutase. Isomerase. These enzymes catalyze the transfer of group within molecules to yield isomeric forms of the substrate. An example is the conversion of fumaric acid to malic acid in the presence of fumarase enzyme. On number 6, we have ligase. These enzymes link two molecules together through the breaking of high energy bonds. For example, acetyl SCOH carboxylase and succinase thiokinase. Properties of enzyme. So we have discussed about the enzyme and the classification of enzyme. Further, I am going to discuss about the properties of enzyme. So number 1, we have the specificity. The specificity of enzyme. Enzymes are specific in their action or function, which means that an enzyme will act on only one substrate or a group of closely related substrate. For example, hexokinase catalyzes the conversion of hexose like glucose, fructose, and mannose to their 6-phosphate derivatives, but glucokinase is specific for glucose only. So it was all about the specificity of enzyme. On number 2, we have protein nature. The nature of enzyme. Enzyme with few exception are protein in nature. They are produced by a living cell but act in vivo as well as in vitro. On number 3, we have direction of enzyme reaction. What is the direction of enzyme reaction? Most enzyme reactions are reversible. The same enzyme can catalyze reaction in both directions. Isoenzyme. These are the enzymes from the same organisms which catalyze the same reaction but are chemically and physically distinct from each other. Factors affecting enzyme activity. So these are the factors which affect the enzyme activity. So it was all about enzyme, the classification of enzyme, oxidoreductase, transferases, hydrolases, lysases, isomerases, and ligases. These are the all are the types of enzymes and the properties of enzyme in which we have discussed about the specificity, protein nature, and the direction of reaction of enzymes. So this was all about today's video. Further I will discuss in the next upcoming video. So please like, subscribe and share my channel and press the bell icon so that you can get all my new upcoming videos.